Hi everyone, thanks for watching and welcome to Past Physiology Review of Topic 1. We'll be focusing on the extracellular matrix, notable in quantity in connective tissue. Okay, so we're really talking about connective tissue here. A quick review of connective tissue. The cells are loosely dispersed within the tissue. Between the cells we have the extracellular matrix. The extracellular matrix is composed of various fibers and this gelatinous goo medium called ground substance. We'll cover these two things in detail going forward. And lastly, talk a little bit about interstitial or extracellular fluid. Please note that connective tissue is highly vascular, thus the red color of the row. And we'll be looking at our textbook, Chapter 5 of the 13th edition of Junkiera. Let's look at Table 1 and Figure 2 to get started here. This is generally what we're talking about. When we're looking at connective tissue, we've got fibers running through it. We have ground substance. This little box right here shows that this kind of pale color, whatever this background color is, okay, whatever you see this color to be, periwinkle, I don't know, pink, light pink, rose, all this stuff, everything between the shapes and the lines is ground substance. It is water, ground substance is water bound up to sugar, essentially, okay, along with electrolytes like sodium and potassium. We see various fibers. We see elastic fibers and collagen fibers and reticular fibers formed of collagen as well. We see blood vessels running through the tissue. We see cells. And we see a variety of cells. These different shaped blobs are different cells. You don't need to know too many specifics about the different types of cells for this, but we will mention them a little bit so you have a base understanding when you come across these terms in class and as you're reading. So, the extracellular matrix in the picture should match up with our text over here, right? We've got ground substance, fibers, okay, protein fibers, elastic and collagen fibers. They're all formed out of protein, okay? So they're all fibers. And then the other component here that's not in this illustration is interstitial fluid, which is in connective tissue. But please note that extracellular matrix does not include extracellular fluid, okay? ECM, extracellular matrix, is just fibers and ground substance, okay? So there are three overarching components of connective tissue: cells, matrix, and fluid, okay? Cells, matrix, and fluid. Cells, matrix, and fluid. And the matrix sounds complicated because it is. It's combined of multiple things. It's a matrix of fibers and ground substance, okay? That's what we're going to be diving into going forward. This table illustrates fiber types, what they are made of. Okay, so the names of the fiber type, the type of collagen fiber that they're made of, except for elastin. Okay, proteins and elastin fibers make up elastic. But basically, this is all what type of collagen makes up the fiber type. And then the locations in the body, where they're found, and what they're like. The last column is the function and characteristics. Notable things about the fiber type. We've got a few memory tools and images and things that we can use to help us recall this stuff. And we've got a clinical note in green at the bottom about vitamin C deficiency that I promise you is going to come up in multiple classes and probably multiple questions on multiple board exams. This concept of vitamin C deficiency leading to scurvy, which is due to impaired collagen formation because of the absence of the coenzyme ascorbic acid, which is a coenzyme for the hydroxylation of proline and lysine. You don't need to know maybe this much detail for this class, but you might as well just learn this now because it's coming up in biochemistry, nutrition, maybe histology, certainly pathology, general diagnosis, okay? All those different classes can ask these types of questions, can ask this question in those sections and boards as well. Okay, so you're going to have bleeding gums and, basically your tissues fall apart. Okay, your tissues fall apart and you bleed. It also is going to affect organs that are high in type 3 connective tissue, filtering type organs, connective or kidneys, spleen, lymph nodes, and bone marrow. Okay, so now that we've got that random clinical note out of the way, we're going to get into the nitty gritty with numbers and names. Okay, so table 5.3 and figure 5.11 are going to show us a little bit about fibrillar collagen. So I'm going to get there in the text right here. Actually, before we get there, here's a little table. You could pause this. You could pause this video at any time, you guys, and read the textbook off the video because there's more details that I'm talking about. The more you read this textbook, the more you learn. But you can also, get lost in more details than you'll be tested on. So, I advise you just kind of for sure look at every table and figure in the book. Look at all the pictures. Look at the captions to the pictures. Anytime you see a word that you remember from class or a term that you see in your textbook, read about it. And if you see a term that is certainly not in your notes, then maybe kind of skip over that just to save time, okay? This table here is not really testable, but it's something that you should know for some background so you know what these words are when you come across them. But this is just about different types of cells in connective tissue. So fibroblasts, okay, anytime you come across this word blast or blastic activity, this means the creation of something, making something, okay? So fibroblasts, blast means to build or to make, okay, that B, just think builders. Fibro builders, they're fiber builders, okay? You can also call them fibrocytes. The word cyte attached to any word just means cell. So cyte means cell. So when you see this word lymphocytes, it basically means lymphatic cell. Okay? So fibroblasts are found in connective tissue. The major product or activity, they make fibers. Okay? Some of them also make ground substance, which we'll talk about later on when we get down here. Plasma cells are B cells that make antibodies. Lymphocytes, they're an immune cell, functioning within the lymphatic system. Any type of leukocyte is another white blood cell involved with immunity. These three are all immune cells. You don't need to know details, but neutrophils and macrophages, they're both white blood cells that gobble up things that are not supposed to be in the body. Phagocytosis means the eating of cells. Cytosis refers to cell. Phage, okay, or phago, this means to eat, okay? Phage means eat, basically, okay? This is all Greek word origins here. Greek etymology. If you ever want to know what a word means, Google the word plus the term etymology. Okay, if I do this, we go to google.com, type in, that's Google Maps. Okay, I'm just going to do it in the bar here. Etymology. The study of the origin of words. Okay, so if you type in any word plus this one, you'll get these different online etymology dictionaries. Super handy, especially if you don't have a great background in medical terminology. This is a great way to learn what words mean, what words literally mean, because a lot of medical terminology is going to be found within different types of words, okay? So macrophages are like a real classic immune system cell that eats things. So they're going to be eating garbage, basically, in tissue, and they also do other stuff that you'll get into in immunology class. They secrete basically cytokines, okay, to make other immune cells do their job, okay, but forget about that. Just know phagocytosis is the eating up of stuff, okay, and neutrophils are going to eat bacteria. Macrophages also eat bacteria. Mast cells are another immune system cell, and adipocytes, okay, store fat. So adipose tissue is fat tissue. Adipocytes are fat cells, okay? This is a nice basic background because some of these terms are going to come up throughout the course, and in all these terms are going to come up eventually. So we might as well just get you off with a good footing, a good start. Okay. So back to where we were going, which is table 5-3 in the text. Come down here. Table 5.3. There we go. This has way more details than you need, but it will have all the different types of collagen fibers plus some, and it's going to have details you don't need, like the size of the structure. You don't need to know the molecule composition. You don't need to know what it looks like under a microscope. But if you want to come back to this table, then you can review where things are found and the major function if you want to, because that can help you remember. That can basically help you remember locations, perhaps. Okay, so something that's resistant to pressure, okay, is gonna, that makes sense that would be in cartilage, which resists pressure, okay, in your joints, and in the vitreous body of the eye. That's something you'll get into later. Resistance to tension, right? Type 1, the major one, is going to be found in things that get torqued on a lot, like skin and tendon and bone, okay, also in your teeth. So I'm not going to go over this, but if you want to come to this later, I've noted it here for you. Otherwise, we're going to go to figure 511 to look at a picture. This is just a picture of a little piece of collagen, a collagen subunit. It is kind of like a rope, okay? It's like a rope made out of protein, all right? We'll keep it that simple. You can get more into detail if you want, but it's wrapped up like this in like a little rope or a little string. And come down to 511, and we've got the larger complex of collagen. It's really basically formed like rope. Rope is lots of little twiny deals wound up, and then bigger strings wound up together. It's basically a bunch of fibers bundled up, okay? So if you want to get into this a little bit more detail, that's what collagen looks like, I guess. But otherwise, we can use this table and kind of focus on this table because I've got the tools for you in here. So reticular fibers are going to be made out of type 3 collagen. And there is a type of fibrillar collagen that will also be made out of type 3. Otherwise, there will be no overlap that you have to remember, okay, as far as numbers go. It's pretty straightforward. Fibrillar collagen, fat 1, 2, 3. Fibrillar fat 1, 2, 3. Conveniently, the biggest, thickest, and most common type of collagen is type 1. It's number 1. It's number 1 most common. It's number 1 most big, okay? Fat 1, 2, 3, these are the big boys. Fat 1, 2, 3, fat fibrillar collagen 1, 2, 3. They are strong, okay? They're big, so that would make sense if they're found in places where you have strong attachments and tough coverings. Strong attachments would be tendons and ligaments, and tough coverings like the dura of the CNS and capsules around organs, and also in your bones. That's not noted in your notes, or they haven't been in the past, but type 1, especially collagen, or fibrillar collagen in general, is going to be found in bones. Now on to reticular. Reticular fibers are going to be comprised of type 3 collagen, and you see in front of you a red tricycle. Reticular red tricycle. A tricycle has 3 wheels. Reticular sounds like red. Everything in here is red, okay? So red tricycle, red reticular, tricycle for 3, type 3 collagen, red tricycle. It's going to be found in basically filtering type organs in the body. And I mentioned etymology a few minutes ago. And the etymology of the word reticular refers to net-like. Okay, and so this is really, really helpful. If you know that reticular means net-like, that's going to help you remember that reticular fibers are going to be found in places where a net would come in handy, like filtering organs like the liver, spleen, lymph nodes, and red bone marrow. And maybe you don't know that that's the function of these, but maybe to keep it simple, okay, blood filters through your spleen, and lymph filters through lymph nodes. Your liver your liver filters, all of your blood and does 499 other things too. And then your red bone marrow, it needs, you need to be able to move red, bone cells through it, and hold back whatever's not supposed to be getting through as well. Okay, so It's a net-like connective or net-like fiber, and so the function could be described as a flexible scaffolding, right? Almost like a net. It allows filtration and easy passage of small things through it. The fibers are very thin. It's a fine net. It's also associated with scurvy, as I mentioned at the beginning. Now on to number 4. The type 4 collagen is going to be handily creating network forming fiber types. So network forming fiber types, there's a thing called a network forming collagen fiber. Okay, I put C here to remind us that this is network forming collagen. Network forming, just say network 4, as in the number 4, and you've got your fiber type. It's comprised mostly of type 4 collagen. And the 4th letter of the alphabet is D, A, B, C, D. Lamina densa is where we find this as a specific special location. The lamina D densa, 1, 2, 3, 4. Type 4 collagen network forming fibers. That all lines up nice and clean. So these are ultra thin fibers in a network type configuration, kind of like reticular, but it's a lot finer, a lot smaller. And you might recall that it's smaller because we come up here. This is a small space here. Okay, the basement membrane is a thin layer between a row of epithelial cells and then the connective tissue below it. So it's small. All right, if that helps you understand it better. Now getting into this purple row here, we talked a little bit about linking, anchoring L7, type 7 collagen, forming the bond between the basal lamina of the epithelium and the reticular lamina of the connective tissue below within the basement membrane. Okay, this is what we're getting into, this link anchor type 7 collagen, L7, right? Linking, anchoring type 7 collagen. Where is it found? It's in the extracellular matrix, linking fibrillar collagens to other stuff. Linking and anchoring, it links fibrillar collagen to other stuff in the extracellular matrix. I suppose that makes sense if it's linking, anchoring, and it links stuff together within the ECM. Okay, that just makes sense. And then in the basement membrane, we just talked about it. It's basically it's the smallest connecting point, right, between the epithelium and connective tissue. So between the basal lamina and the reticular lamina. All right, and it's in purple because it's what is between the blue epithelium and the red connective tissue. All right, so you've got L7, you've got the color purple, and you've got this term, you got the word link. All right, so if you see that word linking things together, linking, anchoring type 7 is a pretty good guess. Elastic fiber types are made out of elastin. This elastin is only going to be found in elastic fiber. Super easy. Probably won't be asked about because it's too easy. It's going to be found in loose connective tissues as opposed to dense and tough connective tissues. That makes sense. These up here, fibrillar collagen, when I'm talking about, tendons and dura and things like that, those are certainly dense connected tissues as opposed to elastic, stretchy, that's loose. Loose connective tissue, also elastic connective tissue and elastic cartilage. Certainly that's where it's going to be found, so I added the word duh here. And the function is elasticity or stretchy, recoil. If we go to figure 15, it's kind of cool to see how these things are arranged. The molecular basis of elastic fiber elasticity. On the top here is what elastic tissue looks like when there's no stress stretching it out. It's a bunch of really kind of random crazy cross links with kind of extra slack basically built into it, so that it's a balled up network of stuff with a bunch of cross links. And then when you stress it out and tug it, it has the ability to unwind out of this kind of weird random coil, type of shape. These cross links, is kind of what we're talking about when we have this vitamin C deficiency. You don't get these proper cross linkings when you're missing vitamin C. Okay, so that's kind of what scurvy ties into. You don't get this proper formation with these strong cross links. So yeah. So anyway, moving on, we're going to get into ground substance, and this can be a bear, but we're going to make it as easy as possible. The text covers it on page 113. I put a big star on it in my PDF version here because it's worth reading. There are a few terms that you don't need to necessarily know, and there are some details like the molecular weight of hyaluronic acid. You don't need to know that weight, but you need to know that it's big. You need to know that it's huge. Otherwise, I highly recommend pausing the video and reading what you can see on here, okay? And better yet, read it in your own book. So what we've got is a table that is 3 rows. These are the 3 big parts of ground substance: glycosaminoglycans, which we just call them GAGs. When I say GAG, that means glycosaminoglycan. Okay, amino, glycan, sugar, protein, sugar. All right, it's basically sugar. It's mostly sugar. It's a huge, long chain of sugar. It's a long, linear polysaccharide. It's really big. It's also the most common GAG, or glycosaminoglycan, in the body. It's super important. Make sure that you really understand every bit of this table. Now, proteoglycans are made of GAGs and a protein. Okay, so what we have is a core protein. I'm going to go to the table here so we can see this while we're talking about it. This is a nice illustration to basically lay out everything that's in here. Okay, so hyaluronic acid on this table is this huge, long polysaccharide sugar chain. Long chain of sugar. It's huge. And then this picture chooses to show proteoglycan complexes bound onto hyaluronic acid. So by itself, hyaluronic acid, or hyaluronan, or hyaluronate, all the same thing. Hyaluronic acid is just this huge, long sugar chain. And then separately, what this is, I'm even going to draw a circle around it. This, and that's way too thick, so I'm going to thin that out. Sorry about that. This is the proteoglycan complex. Okay? A proteoglycan is a protein core, which is blue on this picture. This is protein. It's a protein core. It's a protein rod. And then bound onto the protein rod are a whole bunch of glycosaminoglycans. Bound onto that protein rod are sulfated glycosaminoglycans that I have not talked about yet. Okay, now this is what gives us the bottle brush, quote-unquote bottle brush arrangement. If you don't know what a bottle brush is, just Google it so I don't have to try to explain it to you, but it's a brush that brushes out bottles. All right, so coming back to the GAGs, or the glycosaminoglycans, you have 5 GAGs total that you need to deal with, okay? But you have basically 2 types of GAGs. You have hyaluronic acid, and you have sulfated GAGs, okay? Sulfated GAGs are attached to protein rods to form proteoglycans. There are 4 types of sulfated GAGs that you need to know these words and be able to rattle them off: chondroitin sulfate, keratin sulfate, dermatin sulfate, and heparin sulfate. Be able to rattle these off and list them, because you probably have to list them for a test question, or at least know them. Okay? Chondroitin, keratin, dermatin, heparin, sulfate. These are the sulfated GAGs. There are 4 sulfated GAGs: chondroitin, keratin, dermatin, and heparin sulfate. These, what they do is they get bound up to protein rods. When they are bound up to a protein rod, they form these proteoglycans. You could call it a proteoglycan complex if that helps you remember that it's kind of, multiple things going on. It's a whole bunch of sugar and a little bit of protein. So in gray, I put that more sugar than protein. So now coming over here to the function, what the proteoglycan does, essentially it binds water. So do gags, though, okay? So gag on salt water is our thing here. If you've ever taken a gulp of salt water swimming in the ocean, I bet you gagged, and if you didn't, there's something wrong with you. Okay, salt water is nasty. So you gag on salt water. Glycosaminoglycans is just what they're doing is binding up lots of water along with salt. Salt follows water in the body. Salt and water go together. Okay, so what gags do is they bind sodium and potassium and therefore water. They're incredibly hydrophilic. Know this word. Philic, okay, going back to Greek etymology, philic or a phile with a ph, that means that you like something, okay? Like a pedophile, okay, means pediatrics is children, right? Peds, kids. Phile means to like. So pedophiles, right, are sexually attracted to children. That's what those words mean, okay? Hydro means water, of course, right? So hydrophilic means it's water loving. So gags love water. Gag on salt water, though, because more specifically, they're binding sodium, potassium, and water. You could just say electrolytes and water, but you should, know that it's sodium and potassium especially. So what happens then is that by binding up all this water, they're kind of making a water bed. They're making a cushion. And it's not just straight water, really. When it binds up all this, when sugar binds this water, think of it making kind of like syrup. Okay, if you've ever made simple syrup, working behind a bar or something, it is, simple syrup is just your hot water with sugar added to it, and it makes that kind of a little thicker than water because the sugar is, dissolved into it, okay, bound into it. So, think of a water bed. Gag on salt water, electrolytes and water fill up this space with something like a water bed, but it's a thicker type of a medium than water itself. And so then essentially this cushion, this gelatinous goo, this simple syrup type of mixture is going to be binding and containing basically everything in extracellular matrix. And it's also going to have the function of, allowing some diffusion of products and nutrients through it, and it's going to be pushing out and keeping out bacteria and stuff like that as well. These gags also, bind fibers and cell integrins, and cell integrins are just basically little things that connect the inside of a cell with the outside world of a cell. Okay. So coming back down here to a proteoglycan. Proteoglycan is the protein core with a bunch of sulfated gags attached to it. There are 4 sulfated gags. This table only shows you 2, okay, because different proteoglycans in different parts of the body may only have 1 or 2 types of sulfated gag in that proteoglycan complex. And then this shows you chondroitin sulfate, keratin sulfate, and then hyaluronin. But notice that this arrow, okay, is pointing over here to this. So this is hyaluronin, okay? And then this says sulfated gags, but there should be a little arrow here. Let me draw you an arrow. Sulfated gags, these are sulfated gags, all right? And then this is hyaluronin, okay? So keratin sulfate, maybe this is keratin sulfate. And maybe this one is chondroitin sulfate, okay? So now, coming back to the proteoglycan, it's this bottle brush. The bottle brush, the handle of the brush is the protein. The bristles of the brush are sulfated gags. What are the sulfated gags? List them off. Get fluid with this. Be able to say these words: chondroitin sulfate, keratin sulfate, dermatin sulfate, heparin sulfate. Those form proteoglycans. And what proteoglycans do is essentially they attach gags to bind up lots of water to create ground substance. GS component, okay, so the word ground substance, GS, right? This whole table is ground substance. And notice that the first 2 rows are blue. Think about that. Why would I make glycosaminoglycans and proteoglycans blue? I made them blue because the whole concept is that they're binding up lots and lots of water. Gags and proteoglycans bind up lots and lots of water. Proteoglycans have gags in them. gags bind the salt water. Proteoglycans are a bunch of gags, but just connected by a protein. Actually, I should say proteoglycans are sulfated gags stuck together on a protein. So they're all binding water because gags bind water. Gag on salt water. Proteoglycans also bind signaling proteins, thus the little phone type of signal thing here, just giving you some type of picture. And specifically fibroblast growth factor. So connected onto these, use your imagination. Somewhere you have some little thing, okay, a little protein basically attached to a proteoglycan, in different parts of the body. And if this proteoglycan falls apart due to, just decomposition, tissue death, abnormality, whatever, if Mr. Proteoglycan falls apart, then that is going to make something fall apart that connects to fibroblast growth factor. So basically, fibroblast growth factor is going to know when Mr. Proteoglycan fell apart because he's attached to him. And so if Mr. Proteoglycan falls apart in the body, Mr. Fibroblast growth factor is connected to him, and he goes, okay, I'm going to build some more fibers and start rebuilding this tissue. Okay? That's what this is about, signaling proteins. Now coming over here to this example, you guys, please understand that the examples, okay, column here is talking about like literally examples of names of, okay, the thing on the left side. So examples of glycosaminoglycans are hyaluronic acid, the big one, the most common in the body, the biggest gag in the body. Also, I kind of skipped over this, but this is essential for basically lubrication in joints. If you see something about a joint in the body, we're probably, you got to be looking for hyaluronic acid in your answer choice, and also potentially chondroitin sulfate is associated with joints as well, but especially hyaluronic acid. So those are gags, and then aggrecan and syndecan are proteoglycans. Okay. Sometimes in the notes it can be a little bit confusing because you don't quite get it, but aggrecan just is a type of is a proteoglycan. Syndecan is a proteoglycan. They're just different arrangements. Aggrecan might have dermatin and keratin, and syndecan might have chondroitin and heparin. Okay, I'm randomly just throwing out ideas, but that's what this is talking about. And if you think about it, proteoglycan, aggrecan, proteoglycan, syndecan, they sound like the word proteoglycan. So if that helps you, proteoglycan, aggrecan, syndecan sound the same. But you say, wait, glycosaminoglycan. Wouldn't that be confusing because glycosaminoglycan ends in can? So I don't want to just remember can, aggrecan. I could get that confused with glycosaminoglycan. But no, you won't, because you know the gags. You have to know the gags, okay? You know that the gags, there are 5 gags. There are only 2 types of gags. There are hyaluronic acid and there are sulfated gags. Hyaluronic acid and the sulfated gags are chondroitin, keratin, dermatin, and heparin sulfate. So you're not gonna make that mistake because you know the gags. Okay, you might not remember the names of the proteoglycans, but you can guess because you see can, proteoglycan. Now, coming down to glycoprotein, this whole row is white because I call glycoprotein glucoprotein. Glucoprotein. It is mostly protein with less sugar. Come over here to the picture. A typical glycoprotein. It is a globular protein arrangement with a little sugar attached to it. And it binds things together. It sticks things together. It binds fibers and it binds cell integrins. And if we go to 519 in the text here, figure 519, I'll show you what I mentioned before, what a cell integrin is. I wish this showed the whole cell. But so this middle thing here is the cell membrane. So really, if you think about it, I could put a big circle in here to kind of show you like really there's a huge cell that it's not showing. Okay, so pretend I'm going to do this. I'm going to take the time to do this, make this a little bit bigger. this even bigger. How about that? Okay, so this circle is the cell. Okay, it's a lot bigger than this. And in the circle is in the cell. Try to zoom in. Zoom in without freezing my computer. Okay, so the circle is a cell, okay, to give you an idea. The book shows you the cell membrane because they're zooming in on specific stuff that you don't need to know. But the integrin is just this thing right here. It's just this rod, just this protein rod that goes from the outside of the cell to the inside of the cell. And this is extracellular matrix out here, okay, which has attachments via GAGs and glycoproteins, okay, and the integrin tells the inside of the cell what's going on in the outside of the cell. That's what it's doing, okay? It's integrating integrin. It's integrating the inside of the cell with the outside of the cell. Okay, in the whole idea of this cell is within the extracellular matrix. So there might be fibers and stuff attaching to it, and there's all this is ground substance out here. This is your cell, integrin. Integrin integrates the cell with the outside of the cell. Okay, now examples of glycoproteins. There's only a couple that we've got listed here that you should know about, but it's laminin and fibronectin. And they both sound sticky, don't they? Well, let's break it down. Laminin, okay, what laminin does, it binds epithelium to fibers in the lamina lucida of the basal lamina. Okay, well, at least you can get back to the idea that you can hopefully recall that the laminin is a glycoprotein, because a glycoprotein is a glucoprotein, because a glycoprotein sticks things together. It binds fibers together. It binds cell integrins together. It binds fibers and cell integrins. So laminin, if you think about when you laminate a piece of paper, like your elementary school teacher laminated just about everything she possibly could, when she did that, she just really essentially using by hand or with a machine, she was sticking two pieces of plastic together on both sides of a piece of paper. So when you laminate, you stick the plastic together around the paper. Laminin is sticky. So is fibronectin. Fibronectin. What do hummingbirds drink? They drink nectar, right? They want to drink the nectar of flowers. And the nectar of flowers is sweet. It's got sugar in it. And if you have ever made, like hummingbird feeder stuff, it's like sugar water. And if you make simple syrup type of sugar water, it thickens that and it's sticky. If you were to spill nectar, it would probably be sticky. Fibronectin, fiber, sticky. It binds collagen to the integrin of cells. So those words sound sticky. Laminin, sticky. Fibronectin, sticky. What's sticky? Glue, glucoprotein, binds fibers and cell integrins. And then also this laminin is going to be binding epithelium, basically binding the lamina lucida, okay? So those are the components of ground substance. It's essential that you spend enough time to really get a grip on this. And if you use these simple memory aids and stuff that I've talked about, it should help a lot. Think of your own as well and share them. Otherwise, we're going to wrap up with the recap of ground substance, GAGs, and glycoproteins, and then a little bit about interstitial fluid. You don't need to know much about it. Okay, ground substance, it's clear, but it's thick and more viscous than water. It physically supports cells and fibers within the extracellular matrix. It also regulates cell-to-cell communication. Nutrients can pass through and waste can get out of, slowly via diffusion through. It's not just dead space. There is some transportation of materials through it. And also it does impede bacteria, having access to cells and whatnot. And specifically GAGs that bind up electrolytes and water, make this a thicker type of gel that will cushion, make a waterbed kind of, right? While binding up all this water, can also help block this pathogen invasion. And then glycoproteins are the glue that glue fibers to other fibers and fibers to cells. And integrins are just integrating the inside of the cell with the outside of the cell, like we just talked about. So lastly, interstitial fluid. is the third component of connective tissue. Let's look at it again in red here. Cells, ECM, and interstitial fluid. If you get a question that says, what are the three main components of connective tissue? Then it's cells, extracellular matrix, and fluid. Cells, matrix, and fluid. But if the question is, what are the components of extracellular matrix? Well, then it's fibers and ground substance. Okay, there will be cells within, between the extracellular matrix, but fibers and ground substance make up the matrix. They form the matrix. All of these types of fibers and all of these types of ground substance components form the matrix. So lastly, again, that third component of connective tissue, interstitial fluid. What it is a plasma, quote unquote, right? Plasma filtrate fluid in connective tissue that is free, and it's not bound up by gags or gags within a proteoglycan complex. It's free fluid. That's super important that you know, because we talk about fluid so much. Gag on salt water, right? They're binding up all this water. Interstitial fluid is different. Interstitial fluid is free. It is not bound up. So what it is it comes from your blood plasma. It's just a less protein rich because it's less protein rich than blood plasma because it has to squeeze through your blood vessel walls, and those little fenestra windows, that you learn about later, the little pockets that this blood plasma squeezes through allow fluid to get through, but it's going to be filtering out protein because protein is a lot bigger than the fluid. So normally you have very little interstitial fluid. If you have too much, it's called edema. There are many different causes, and usually it's not a good sign. There are many different pathologies that cause edema. It could just be protein, or it could be trauma, burns, various vascular disorders and things. So just know that edema is a clinical term for excess tissue fluid or excess interstitial fluid. Okay, this is a little diagram kind of showing you what I just talked about. And in your book, shows you a little more complex diagram of blood flowing through the arteriole, going into the tissues, into your capillaries, these tiny little vessels, and then it gets returned back to the heart via venules, which feed into veins. And in here, due to different pressures, this this plasma part of blood can get squeezed out into the fluids. Okay, so that's it for topic 1. Look over what you are not solid on, and I'll see you guys in past class. Find the book, get the book on paper or PDF. It's a really good textbook, and it's certainly worth buying this book, especially a used copy for $20 or something on Amazon. Okay, highly recommend it. Thanks for watching.
is the one.